Hello everyone, this is the third session if you recall, and this session will be on the implantable MEAs. So, third session on the implantable microelectrode arrays. If you recall the second and the first session, then you will remember that we were talking about epilepsy as a model, and in epilepsy we were talking about rodent, because animal model would be rodent, the disease would be epilepsy, and the idea is to acquire the signals from the brain, and those signals we called as a ECOG signals or electrocardiography signals. In the first session what we have seen is we have a flexible microelectrode array with 32 recording electrodes, and we placed it on the rat's brain, and then we can create epilepsy by using the convolutions like bicuculin, and then we can recover the baseline by using different anti-epileptic drugs, and from that we can see what is the efficacy of those anti-epileptic drugs or AEDs, right. Then in the second version what we have seen is that how about we fabricate a bioresorbable flexible microelectrode array, and the application is still same, which is to understand the efficacy of drugs. In that we have seen that once you implant the device and you record the signals, after few days the device will absorb, or it is biodegradable, and in the experimental procedure we have seen that how we are using it in the in vitro situation or in the lab situation, where we place the device in the pH 7.4 and look how it is degrading across period of time. We have done the same experiment on the animal model, which is by implanting the device in rat's brain and looking at how it is degrading across period from 3 days to 7 days. At this point in time we were not having access to the animal MRI, otherwise we can also look whether this in terms of the MRI image how the chip is degrading across period of time, like 1 day, 2 day, 3 day and so on. So, the alternative way of what we understood whether it is absorbing or dissolving in the brain is by looking at the signals that we were able to acquire on day 1 versus day 3 versus day 7. Once the signals are gone, we assumed or we presumed that this is the way to look at the methodology to look at the bioresorbability. We assumed that it is dissolved, but a better way would be to look at the animal's brain in animal MRI and see how the device is dissolving with respect to time. This is the third session where we look at the implantable microelectrode array for recording neural potentials from rodents, and in this case we will be talking about microneedle based microelectrode array, right. You all know about needle and you all know about microelectrode array, how we are integrating those electrodes, recording electrodes onto a single shank or a microneedle electrode. Let us see that. So, as usual, right, we start with the background, we understand the research goals, we look at the objectives, and then we look at the novelty factor. The first is background. Background, as you can see, that though the planar ECOG electrodes, that is you can place on the surface, can provide a better spatial resolution than EEG signals. It offers a limited understanding of the activities at a brain depth where the signal originates, right, and that is very important, like, because you are looking at the surface and acquiring the signal. What happens in the deep down the brain, we do not know, but if we have the microneedle arrays, you can integrate those or you can place those in depth of the brain and can understand or acquire the signals, so that you can further study those signals. Now what we call is as a local field potentials recorded by depth microelectrode arrays are used for studying electrical activities of the brain from cortical columns. Very important term came now, which is cortical column. So, the array of cortical columns, right, you can see the cortex array of cortical column, and each column if you just take it in the brain, right, each cortical column will have 6 layers, okay, 1, 2, 3, 4, 5 and 6, alright. So, second, third, fourth, fifth and sixth. Okay. This is a column, right. You can look at this, it looks like a column. So, this is a column. Now, we are talking about rat or rodent model. Okay. Now, fourth, the layer fourth is an input layer. Layer 1, 2, 3 is processing layer. Layer 5 and 6 are output layer. All right. So, if you want to go inside the brain, deep inside the brain, we need to create a electrode that can fit in this particular column and can measure the changes from each column. What changes you can measure or acquire the electrical signals. Now, to penetrate it, we make it like a needle. All right. So, this each electrode would be a recording electrode. We will see how to fabricate those recording electrodes, and each electrode or group of electrodes should fall with the same within the same layer. So, to acquire signals from each layer. All right. So, can be one electrode, can be two electrodes. So, if there are two electrodes, right, like this, then how many electrodes we require? We require 12 electrodes, right, because 2 per layer, we have 6 layers. So, there are 12 electrodes. So, your device should be like this, where it has 12 electrodes, and then from the brain we can acquire the signals from all these 12 electrodes. Okay. Or if you want to be easier, you can have only 6 electrode, 1, 2, 3, 4, 5, 6, and from each layer you can have one electrode that will acquire the signal. Okay, this much easy. So, what we understand is that now we need to understand the local field potential at different depths. Depth is from layer 1 to layer 6. Okay, great. Now, let us move to the next point. The next point is that the baseline LFPs, baseline local field potentials, local field potential during an induced seizure when the epilepsy is going on, and once you load the anti-epileptic drug or you administer the anti-epileptic drugs, the recovery of the signals, this all things creation of the first is a baseline, then the ECOG during the induced seizure, and the signals that you obtained when the recovery has been occurred. So, this all needs to be studied or to understand the evoke response and effect of anti-epileptic drug on LFPs at different depths, correct, because then only we will know that how the AEDs are effective or not. So, you still understand that the goal is still epilepsy, but in this case we are looking at the depth of the brain rather than the surface by using the depth electrodes. So, what should be the research goals then? The research goals are following. The first one is to record a baseline electrical activities which induced ictal and interictal discharges from depth of a brain in animal models, right. That should be the first baseline electrical activity, and once we can do that, for doing that we need to anyway fabricate a device. Second is that the spatial temporal analysis to determine the changes in electrical activities in different neurological conditions and distribution across the depth. That is another thing that we need to identify or measured or acquire. Now, this changes in electrical activity you can again understand once you have the microelectrode array, and in this case we have to go deep down. That is why I told that we are going to use the needle kind of electrodes. So, in terms of objectives, one is the design and fabrication of microneedle electrode arrays MEA to record the local field potentials normal with ictal and interictal signals during the seizures and during the after the administration of AEDs. The implantation of the array in animal models, right, how to implant it, where to implant it, how deep it should go, that is another thing. The next thing is recording and analysis of LFP signals, local field signals in response to convolution and AEDs for studying the epileptic activity. Now, this is the 3 different objectives. Let me go to the novelty factor. The novelty factor is that fabricated implant device was used to study electrics in normal, epileptic and recovered conditions, but is there any change in the design that we have made which can fall in the novelty. And of course, there was before what we proposed or what we have done or how we have acquired the signal, this open Cyton daisy biosensing board was used only for acquiring signals from human brain. It was never tested on animal models. So we tried it to acquire the signals from the rat's brain. Here is the figure, right. You can very clearly see a beautiful schematic where how we are going to perform the experiments. So, the microneedle that we are talking about has many electrodes that you can see here, about 13, but I told you, right, that there are 6 layers in a column, 6 layers, and why there are 13 electrodes? If I have 2 per column, right, then I should only have 12 electrodes, right, 2 per column, 2 into 1 column. How many columns were there? There are 6 columns. So, 2 into 6 becomes 12, right, simple math, but here you see there are 13 electrodes. So, now can you tell why there are 13 electrodes, right? Think about it, why there are 13 electrodes instead of 12 when we require only 12. If you know the answer, it is great. If you do not know, it is absolutely fine, right. We require 1 electrode for ground and also the remaining. So, with respect to that electrode, we are going to measure the signals, right. So, 1 extra electrode is there, and now the size of this particular needle, right, the depth that it can go is 3 millimeter. You can see the depth, this depth is from this one, this depth, right. Now, why 3 millimeter? Because most of the time the length of this cortical column is 2.5 to 3 millimeter in rat's brain. What is the length? 2.5 to 3 millimeter in the rat's brain, and that is why we have the length of this needle as 3 millimeter. What is the width of this chip? 4.4 millimeter, 3.6 millimeter. This is your micro electrode array, but in this case it is needle which the MEAs or electrode array recording arrays are integrated onto the needle, alright. Now, we have this particular stuff and this is nothing but your micro manipulator, okay. This micro manipulator we can integrate the microneedle, as you can see here. We perform the craniotomy and then you place this needle inside the brain of the rat, okay, inside the brain of a rat. It is micron precision, alright. So, we can control it with the help of the micro manipulator controller, which you can see in this case here, right. Micro manipulator, I already told you that this is shown here. Then microelectrode arrays are here, the zoom in version is here, and then rat stereo adapter is right over here, right. So, we have everything ready. The signals that you acquire goes to the Cyton biosensing board, Cyton daisy biosensing board, and these are transferred to the OpenBCI USB dongle via Bluetooth, right, and what you can see on the screen are the local field potentials from the baseline when there is epileptic activities and when there is a AED that is administered in the rat's brain, right. So, administered in the rat's body. So, the efficacy of AEDs can also be again measured using this particular technique. So, if you read, it is an illustration of the system to record local field potential using microneedle, right, electrode array implanted in the anesthetized rat. It is under anesthesia. The inset shows the design of the implanted MEA. Number of shanks are 1 to 13, represent electrode using for the LFP acquisition. So, the favorite part. What is favorite part? A fabrication, right. So, let us see how we can fabricate this particular needle. Now, what is this particular stuff? You see here, the gray color is silicon, green color is SiO2, gold is TiAu, and blue is oxide, right, just to represent it, oxide, which is by PECVD, right, plasma enhanced chemical vapor deposition. This is a technique by which you can deposit the silicon dioxide or insulating material like silicon nitride and silicon dioxide at a lower temperature. The reason of having lower temperature I will tell you in just a few minutes. Okay. So, first you take a silicon wafer. Now, this everyone understands very well how to fabricate device, isn't it? Then you take and then you grow silicon dioxide on silicon wafer with help of the thermal oxidation technique. So, you can grow silicon dioxide, you can see here, and we have it here. So, what is it? SiO2. Here also we have SiO2. This is silicon, right. So, then we have this particular A, isn't it? Now, what you do is, once you have this one, then you deposit your titanium and gold, because chrome is known to be toxic for the implantable devices. That is why we go for titanium gold. This is again your SiO2, silicon, and then on silicon you have a layer which is nothing but your titanium and gold, TiAu. Okay. So, this is your B now. Okay. So, where is it? This is your A and this is your B. Easy everyone. Now, what we do? We pattern this, you see, we pattern this. So, patterning everybody knows, right. I am just showing you to some cross section of this electrode like this. Okay. So, do not worry about how exactly the top view looks like. This is the side view or cross sectional view. Okay. So, you have your SiO2 and you have your titanium gold, and now I have to pattern this. So, I will spin coat my photoresist. Our favorite photoresist is positive PR. What is this one? TiAu, titanium gold, SiO2, silicon, SiO2. Now, on this we have the mask. We have the mask. Just to represent the electrodes, we will just draw like this, and this will be our bright field mask. Okay. This is our bright field mask. So, this one will be your bright field mask. After that, so positive PR you have to do soft bake, load mask, and then expose with what is this one? UV light. Okay, expose with UV light, and after that you unload the mask and develop the wafer. Unload the mask and develop the wafer. So, what develop the wafer means? Develop the photoresist on the wafer. Okay. Then what will I have? I will have my oxide, and then on my oxide I will have gold, titanium gold. Titanium is used to improve the addition of the gold, and then I would have my photoresist in these two area. Why? Because positive photoresist, the same pattern that is there on the mask will replicate on the wafer or on the photoresist, or in other terms the unexposed area will get stronger and the exposed area will get weaker, right. So, this is my positive photoresist, right. Remaining things you know, TiAu, SiO2, silicon, SiO2. Alright. Now, what I will do? I will dip this wafer, in AU etchant, right, then rinse it with DI, then dip the wafer in titanium etchant, then rinse it with DI and then dry it with nitrogen, dry it with nitrogen. So, first gold etch, because gold is on the top, then we rinse it with DI water, then we dip it in titanium etchant and again rinse it with DI water and finally dry it with N2. Okay. If you do so, what you will get? If I do so, what I will get? Like this, is not it? The area which is protected by the positive photoresist, right, will not get etched, the area which is not protected will get etched, simple. Now, the next step, you dip this wafer in acetone, alright. If I dip this wafer in acetone, then my positive photoresist will be stripped off, right, photoresist will be stripped off and I will be left with my titanium and gold, is not it? I am left with my titanium and gold because my photoresist has been stripped off. So, if you have this, then this will be your C, this one. I hope we all are understanding and we are all on same page, right. So, for A, it is an oxidized silicon wafer using thermal oxide. For B, we have titanium and gold. For C, we have performed a photolithography technique to pattern in the design that we want, which is our microneedle array. After this, the next step, so I will just draw it here so that we do not miss what we have to show. The next step is to grow silicon dioxide using, so I will just show some different symbol for silicon dioxide. Let us say like this, right, this is your silicon dioxide, but why I am drawing like this and not the pattern which is similar to SiO2 like this one, right. So, because this silicon dioxide is grown using plasma enhanced chemical vapor deposition, alright. Now, let us understand why we have to use the oxide which is deposited by PECVD and not thermally grown. In this case, why I have used PECVD and not thermally grown oxide? The reason is that for thermally grown oxide, for thermal oxidation, we all have understood and seen that the temperature is about 900 degrees centigrade to 1100 degrees centigrade, right, and this is extremely high temperature for the material which is below it. That means, in this case it is TiAu, but if it is aluminum, it is nickel, if it is nichrome, right, or some other material that cannot withstand this temperature, then it will then we cannot get our device, right, we cannot get the pattern, the metal will be affected, the material will be affected, and to avoid that the affection of the material, the because of the chemicals or because of the temperature, in this case that is 900 to 1100 degrees centigrade, we go for an alternative technique of growing. So, this 900 to 1100 degrees centigrade is for thermal oxidation, and we have wet oxidation, we have dry oxidation. In both the cases you require this temperature, right, but we cannot have this temperature because the metal below it may not be able to withstand, the material on which you are growing this silicon dioxide may not be able to withstand. So, what is an alternative technique? The alternative technique is to use plasma enhanced chemical vapor deposition to grow your oxide, because PECVD you can use 100 to 300 degrees centigrade to grow your SiO2 or even silicon nitride. You got it now, the reason of using oxide deposited by PECVD? Yes, but if there is no metal, if there is no material, right, then you can grow for the thermal oxide, because thermal oxide always has a better quality of oxide compared to PECVD oxide. Okay. So, after this, now once you have this, what is the next step? Next step, if you see on this, is becomes very easy, that once you have this PEC oxide, which is your D, this is your C, and then now it becomes D, that means that PECVD oxide is there. What is the next step? We have to open the contact pads and open the recording electrodes. You can see here in this microneedle you can see dots, right. These dots are, so let me just try to zoom in further. I do not think I can zoom in, but let me try. Okay. Okay. You see here, now you can see very clearly, right. What you can see is that the oxide from the recording electrodes, right, from the recording electrodes and the contact electrodes. So, you can see my pen moving. These are the contact pads, and these are the dots are the recording electrodes. You can see that blue color is gone from there. That is because we have performed a photolithography technique to remove the oxide only from those electrodes, right, contact electrodes and recording electrodes. Now what you do is you save the front side, and by protecting all the region except this needle region, you see, protecting only needle region, except needle, everything is exposed, that means that only on this region and on this one you would have your photoresist. So, in this case I will have a photoresist which will cover my needle and this part like this and everywhere here. Okay. I will coat photoresist and pattern it using lithography like this. Okay. Now, if I do this and the photoresist is only in this area, the next step what I do is I will etch silicon dioxide. Silicon dioxide etching can be done using RI, reactive ion etching, or using BHF, buffer hydrofluoric acid. If I do that, then I can expose my silicon, isn't it? I can expose the silicon. The next step is I will use DRI. Once SiO2 is removed, I will go for DRI. Deep reactive etching, it will etch the silicon completely from all the area which is not protected by the photoresist. If you do that, then what you get is F, which is here, right. After DRI you get this one, and also this is just a representative like I said, but in reality, right. So yeah, so once you etch completely, right, you catch completely, then you get this one. Now why we do not have to do front to back? Because the wafer that we start with is only about 100 microns. The wafer that we start with is only 100 microns in this case. So the thickness of the cantilever can be 100 microns. Okay. Sorry, not this is not a cantilever, I am sorry, microneedle is only 100 microns, right. It looks like cantilever, but here it is not released, right, it is thick. So it is a microneedle. So microneedle, the thickness is similar to the thickness which is here of the silicon, and like I said, it is 100 microns. Why the thickness 100 micron is used? Because when you go for this needle, right, and then you implant it, then this one would be somewhere around 200 to 300 micrometer. The diameter of the column would vary from 200 to 300 micrometer. 100 micrometer will go in and can record the signals. That is the reason of using 100 micrometer thick wafer. So that finally the microneedle thickness will also come around 100 micrometers. The G shows the actual microelectrode arrays or the electrode arrays on a microneedle, and this is the zoom in version of the same, right, where we can see that how the very clear titanium gold patterns can be seen, right. right, everything is covered by an oxide except the recording electrodes. Now you will say that I cannot see the oxide. You cannot see the oxide because the oxide is about a micron or so, and or even less, like half a micron, and oxide is always transparent, right. So, silicon dioxide, since it is a transparent, you can still see the as if there is nothing on the other area, but all this area actually has oxide, okay, and only the recording electrode and the contact electrode does not have any oxide. This is an SEM of the same, right, SEM of the microneedle. Now, once you implant it, you need to also attach it to the electronics. You can see here, right, this needle is there, but finally it is attached to the electronics so that it can be further used for acquiring the signal. So, for that we have a PCB for wire bonding the fabricated microneedle, right. You can place the microneedle in this area, right, and then it goes like this, and the pads of the microneedle can be wire bonded onto this like this, okay. Alright. So, that is how the PCB can be utilized, and this comes under packaging, okay, electronics packaging. The B is interfacing adapter PCB to interface the signal acquisition system. So, A and B's are very clear. Like any other electrodes that we have fabricated till now, we also have to perform the characterization of this particular micro electrode array, microneedle electrode array. So, for that we have done the experiment using electrochemical impedance spectroscopy, and the device is attached to the micromanipulator arm, as you can see in the figure A. While the impedance spectroscopy data is shown in figure B is impedance versus frequency plot after immersing the recording area of the fabricated device at pH 7.4. In this case, what you can see is that an actual image or photo of the rat during the experiment, and you can very clearly see that how the acrylic arm is connected to the micromanipulator, to which there is an FFC cable attached, there is an FPC connector, and to which there is a fabricated MEA wire bonded to the PCB, and there is a somatosensory cortex on which the device needs to be placed or implanted, and then there is a cytone daisy board. Now, in this case the B shows that we are ready to implant, the C shows that the needle is inside the rat's brain. So, when you record the local field potentials, how it will look like, alright. So, you can see that the recorded channels are 13. One, as I told you, with respect to the channel we keep on recording the signals. So, the y axis is baseline microvolts, also the number of channels, and the in terms of voltage, and then on the x axis you have time, and its time is for 20 seconds. So, you can see that how the voltage would vary with respect to time, and in case of baseline signals the maximum amplitude is 50 microvolts, while in case of epileptoform discharges you can very clearly see that the signals has the strength has increased to 500 microvolts in that range, while in case of administering the AED through the rat's tail, what we can observe here is that the baseline is recovered and the amplitude value is close to 50 microvolts. When we perform the time frequency analysis, you can see that when we get the baseline values, and as you can see from A1, A2 and A3, right, A1 shows the LFPs baseline, then A2 shows the power spectrum analysis, and A3 shows the corresponding frequency with respect to time. The power spectrum analysis and all this A1, A2, A3 will show the baseline form. As soon as you apply the topically the bacuculin, which is a convolution, and wait for 7 minutes, you can start seeing the episodes of the epileptic activities, and it can be clearly visible also in frequency spectra, power spectra. And when you apply the intraperitoneally the phenytoin sodium, which is the anti-epileptic drug. After 4 minutes the baselines are restored. Here also you can very clearly see the LFPs reflected after administrating the AEDs, and same thing power spectrum and the frequency spectrum. So, this is the histology of the rat's brain. You can see that how the needle has penetrated right inside the rat's brain, and it is a crystal violet assay. The figure shows that a minimal tissue damage due to the in vivo experiments has occurred to this using this particular what do you call the experiment methodology. We have some fabricated, we have some patents and some publications around this area. Right now let us not worry about it. Now let us see the last part of it, which is the last session. Now what I want to show, until now what we have seen an implantable flexible microelectrode array, then we have seen bioresorbable implantable microelectrode array, then we have seen single shank, it is a one shank, right, one needle, one shank integrated with microelectrode arrays. Now in this session what I want to show it to you, that what happens when you want to acquire the signal from multiple regions in the same area of the brain, right. So for that you need to create more than one microneedle. It can be 2, 3, 4 depending on what is the area of interest, right. So for hippocampus the way to design the electrodes would be different compared to the motor area, compared to the visual area and different regions in the brain. So let us see what are the different kind of microelectrode arrays that you can fabricate. Here you see that there is a 3 shank microneedle linear MEAs for studying the spatial distribution of the LFPs, and here the again the depth is close to 3 millimeter. The distance between 2 electrodes or 2 shank is about 200 microns. Each recording electrode, as you can see here, each recording electrode is close to 15 micrometer, and the spacing between each electrode is 25 micrometer. Means that if I zoom this one, then I have this, right. So this is my 15 micrometer by 15 micrometer, okay, and the distance between 2. So I have one more electrode, then the distance between 2 is about 25 micrometers, okay. So that is where we are from the understanding the electrode size, recording electrode size, and the it will record somewhere from 415 micron area. What else? Yeah, the width, this width, right, this width is about 10 microns, 10 micrometers, alright, and the there are 3 masks, is a 3 mask process. So mask 1, mask 2, mask 3. The materials are silicon as a substrate, wafer thickness close to 200 micrometers. Like I said, you can convert, you can change the thickness from 100 micrometers to 200 micrometers. If you really want to have only 10 micrometer thick wafer, then there is an alternative technique than what I have been showing it to you, which will not take as part of this particular course. Otherwise it becomes more like the fabrication and less like the application, right. So now we have the electrodes and interconnects which are made up of AU. When you say AU, there should be always an adhesion layer, which is your titanium or chrome. I told you that chrome is not biocompatible, that is why we go for the titanium gold, and the insulating layer in this case is silicon dioxide. So these things are absolutely clear with all of us. Now let us look at the device specifications. So electrode size is 15 by 15, inter electrode gap is 25, number of electrodes or shanks are 11, right. Is it correct, number of electrodes per shank? Per shank. So let us understand, this is 1, right, 1, 2, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This is 11, this is 1, right, 1 to 11. So there are 11 electrodes on the particular shank, on each shank. The width is about 200 microns, length is 3000 microns, which is 3 millimeter. Distance between the tip and the first electrode is 250 micrometers. The contact pad size is 300 by 300 micrometers, which is the contact pad is this one. Okay, if I have then this one, (300 by 300). Okay, that is the contact pad size here, and with 100 micrometer gap, that means that between two contact pads there is a 100 micrometer gap. This is about 300. Okay, these contact pads. Dimension of the tip is 25 microns, and final angle of the tip is 30.25 degree. So this is what are the specifications of this micro electrode single shank. Now we have 3 shanks here, so we can cover a larger area. This is an actual photograph. As you can see, right, we have placed the micro electrode structure, 3 shank electrodes against one cent to see the size, and you can appreciate that how small these needles are. If you have ever seen 1 cent, then you will really know that this is really tiny. Okay, and then you can see different photographs of those microneedles, including the one here, and if you zoom in, it looks like this, a beautiful. You can see very clearly here is a purple color, right, the purple color. It should be gray if it is silicon, but purple because there is a silicon dioxide that is grown on the gold or deposited on the gold, and then only gold contact pads and the recording electrodes, and so and the contact electrodes and the recording electrodes, which is the these are the recording electrodes. The oxide is removed from this region, right, and then finally the DRI is performed to realize the microneedles. Now in the area of neuroscience, right, we use 4 tungsten wire and fuse it together, right, like this, so that you have on the tip you will have 4 electrodes like this. Okay, these 4 electrodes are at the tip. You can understand like this, the tip is there, and 1, 2, 3 and 4, and these 4 electrodes will be able to measure the signals from the surface or from the depth. These are called tetrodes because they are 4 in number, so they are called tetrodes, and we can have 2 different tetrode on the planar structure. So in this case you can see that there is a device with 4 shanks, and each shank has 2 tetrodes, tetrode 1 and tetrode 2. So we have total 8 tetrodes. Okay. So the materials are silicon substrate, wafer thickness about 200 micrometers, electrodes and interconnects which is made up of gold, insulating layer which is silicon dioxide. Device specifications are again given in this particular case, where the electrodes are of 15 micron by 50 micrometers. The inter-electrode gap is about 30 micrometers, which is the edge to edge. And then distance between quadruple electrodes or quads are about 100 micrometers. Number of electrodes are 8, right, distance this one is 100 micrometers. Okay, 100 micrometers, and then number of electrodes are 8 on each shank. The length of shank is 3.1 millimeter. Distance between tip and the first electrode is about 270 microns. The contact pad size is about 300 by 300 microns with width of 100 micron with 100 micrometer gap between 2 contact pads. This 100 micron we are talking about is edge to edge. Finally, the dimension of the tips are somewhere around 25 micrometer, and the angle of the tip is 29 degrees centigrade. Actual photo, not just simulation or theory, right, it is an actually fabricated device. And when we say fabricated device, it requires you to design, it requires you to design the mask, do the simulation, and then to fabricate. So, these are actual fabricated device done right here in the Indian Institute of Science, using the facilities. We use laboratory facilities that I have in the Department of Electronic System Engineering. Is a nanofab lab with lot of interesting important equipment, and for some of the processes we use the mother facility. So, the mother facility is what I have coined it. We have a nano fabrication facility which is SENSE, and there we use all the CVD and RI techniques. Here we can use PVD techniques, deposition, thermal E-beam sputtering, we can use photolithography, wet etching, optical microscope, and so on. There we use PCVD, LPCVD, RI, DRI, and some kind of etching like SEM, right, and so on and so forth. So, if you see the screen again, we are almost at the last few slides, right, about the microneedles, right. We talk about lot about the epilepsy and brain. So, I am sure that you want to move to next kind of topic, right, but I just wanted to tell you that we are now in the capability of fabricating our own implantable sensors and recording electrodes. Okay. Until now we used to buy, and we still are buying, and we are now trying to make everything within our country. So, that is like I said, is the last slide, and we are finishing the session 4 at this point with a point that these are not just what I am showing it to you and we are done, but I will also show you the recorded video which we have done as a part of the experimental laboratory. And for each video I will first introduce to you what you are going to see, and then we will look into the processes. The next session, right, would be on the electrical stimulation that we are going to use for the application in case of the deep brain stimulation. Deep brain stimulation is nothing but the Parkinson disease, and how we can use this deep brain stimulation, what kind of experiments we can do or we are planning to do, and again everything comes in the advanced stimulation. Now I hope that you appreciate the title of the course. It is not just talking about the EEG signals and ECOG signals and different electrodes, but also to fabricate those electrodes and implant it and record the signal, and simultaneously we are going one step further of applying the electrical stimulation and correspondingly recording the signal, right. I told you about two different models. One is acute experiments, one is the chronic experiment. Acute experiment is where the animal is used and utilized, you can say, because we do not sacrifice the animal without any purpose, right. We take the animal, we implant the animal with the electrodes. Once the work is done, the animal is euthanized, while in the chronic experiment we let the animal live for certain days till we acquire the signal. For everything, whether it is animal model or human model, you need to have a clear ethical clearance to use those many subjects, whether number of animals or on number of human subjects, right, like in the case of the neonatal hearing screening, right. So, with that I will stop this discussion about microelectrode array, but one more topic we will be talking about where we will see that how the electrical stimulation can be used for a very important disease other than epilepsy, which is Parkinson. So, let us meet in the next class and we will talk about that. Till then, you take care, see you then, bye bye.